Good morning, everyone. I'm Ben Butkus, Managing Editor at GenomeWeb, and I'll be your moderator today. Today's webinar is entitled Novel Applications of NGS in Cancer Diagnostics. The sponsor of this webinar is QIAGEN. Our panelists today are Reinhard Büttner, Chairman of the Institute for Pathology at the University Hospital Cologne, and Vikram Devgan, Head of the Biological Research Content Business at QIAGEN. A reminder to attendees, you may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after the presentations have concluded. We're going to start the webinar with Reinhard Büttner of the University Hospital Cologne. Please allow us a moment as we hand the webinar over to Reinhard. Reinhard, please begin whenever you're ready.
Hello, everyone. Good morning and good afternoon, wherever you are. As an addition to the introduction, I should like to say that I am a diagnostic pathologist, an anatomic pathologist who sees slides from cancer patients every day on a routine basis and has to answer the question, what is the best treatment for this patient? And therefore, the question I would like to address today is the question of what would we like to add to mere morphological information by histology and by adding genome analysis. The question here is also what is a suitable alternate therapy for the individual patients. So we have recently summarized this question of changing a mere morphological pathology to a more genomic-driven and genomic-based classification of tumors. So there are 5 areas where genetic or genomic analysis derived from cancer tissue slides will change and modify our diagnostics. This is the basis of classification, what is an entity and a disease in tumors. Then the big area of prediction of response to therapy. This will be very important in many instances. No therapy will also be the best therapy. So we have to predict the clinical course of a disease. That is prognosis. Now, what is will be the course, and what will during the course be the best therapy. And finally, even in the good old tumor diagnostic staging, what is the metastasis, what is the primary tumor, genomic information has a lot to tell and is modifying our way of thinking. And also, I will very briefly touch the question of surveillance, how to monitor a disease under treatment, a tumor disease, and to predict emergence of resistance and relapse. As you can see, this is a summary from our big genomic data published last year of genomic analysis of 6,000 lung adenocarcinoma, and it simply classifies over chromosome from 1 to 22, and this is individual tumors from 1 to 1,200, just gains and losses, gains in red and losses in blue. And by only this one dimension of genomic information, you can immediately classify all adenocarcinomas, all squamous carcinoma, and the group of not otherwise specified unclear tumors are being nicely classified into neuroendocrine tumors, into large cell neuroendocrine and small cell cancers. And besides this, there are oncogenic driver lesions, which are specific for all these tumor entities. In adenocarcinomas, for example, EGFR mutations. In squamous cell carcinoma, for example, FGFR amplification and so forth. So the big entities, let's say adenocarcinoma of the lung, is now falling into a basket of many different entities that are driven by specific oncogenic events and need specific therapies. First, in the good old days, we would simply classify lung adenocarcinomas. But if you subclassify now to KRAS-driven tumors, FGFR-amplified tumors, EGFR-mutated adenocarcinomas, or ROS1 translocated, you can immediately see that these diseases have different prognosis. These are all patients in stage 4 with systemic metastasis. And as you can see, the best survival have ROS1-driven tumors. FGFR is a lesion associated to a very poor prognosis and associated to smoking status. So the genomic classification really has implications for prognosis and for classifying individual subgroups which have their specific epidemiology. And also prediction, of course, is a very big subject. What patients should receive what drug. And for many years, oncologists have been very reluctant to believe that a specific lesion like EGFR mutation and activating of EGFR signaling or an ALK fusion activating the ALK kinase signaling can be treated by a specific inhibitor, and the patient would have an overall survival benefit. The argumentation was, okay, these mutations are prognostic, so you select out only those tumor patients that have a better prognosis, and then you treat them and think it's your treatment. However, we have retrospectively analyzed in our very large consortium around the Cologne area in Germany those patients with EGFR mutations that did not receive a TKI and those that did receive a TKI. And you can see that there's a massive difference in overall survival of 2 years, so you triple survival of these patients. And even in second line, still there is 1 year to gain. However, this is only one dimension, so we now classify tumors in EGFR positive, EGFR negative, ALK fusion positive, ALK negative, KRAS positive, and so forth. But also these patients, when they are treated and live for longer, they travel over time and develop resistance mutations. So they're not cured, but the tumor changes in the genomic composition. So we have to classify for the first diagnosis for every lung tumor that is not treated by surgery, all these lesions to answer the question, can we apply a drug that is approved? Can we apply drugs, the yellow ones, like ROS inhibitor, MET inhibitor, BRAF inhibitors, RET inhibitors? These are available by off-label treatments, so are approved in tumors other than lung, outside, for example, BRAF lesions can be treated in melanoma. And then there are a number of genomic lesions that are in early clinical trials and probably lead to very effective treatments. So this is one dimension, to classify a tumor along the oncogenic driver lesions. And as you can see, this is possible both in adenocarcinomas and in squamous lung cancer. However, under treatment, these tumors evolve, develop resistance, and change their genotype along the treatment. So I have this patient, for example. This was one of our first patients that we diagnosed already 6 years ago at stage 4 with multiple brain metastasis, had a very nice activation exon 90 mutation, and was one of the first long-term survivors under Erlotinib in our cancer center. Then 2 years later, she relapsed, and the brain metastasis started to grow again. We again got a biopsy, and she developed a T790M resistance mutation that makes Erlotinib ineffective to inhibit the EGFR receptor. By this time, a second generation of EGFR inhibitors became available, afatinib, and the combination with an EGFR antibody proved to be very effective, and the patient had another response for more than a year. Then she relapsed again and had a MET amplification. So he had another genomic alteration, and at that time we did not have any further therapeutic options. We of course would now offer her treatment with crizotinib, and I can show you this is the pathway. So the original activating mutation that causes MAP kinase and ACT activation. Then she developed this T790M mutation that prevented the inhibitor from binding and reactivated the original EGFR signaling pathway. And then this was blocked by the second generation inhibitor and the antibody, and then she developed an amplification of this rescue receptor, MET, that sort of signals into the EGFR receptor pathway and restores again the EGFR dependency. And you can see in the third biopsy, we see in the red signals this massive amplification in c-MET, which was not present in the previous biopsy where only the T790M resistance mutation was present. So it is very important to monitor patients and ask the question, is my therapy still effective or do I see emergence of resistance? And of course, today, this patient would be treatable. And I can share this patient that has also an exon 19 mutation in EGFR and had a massive progression here under treatment with Tarceva, developed multiple metastases in the bone, in the liver, heart. And this patient massively benefited from combination with crizotinib, and because this patient had a MET amplification. So depending on the specific mechanism of resistance, we can now deliver at least one, in many cases, two further lines of specific targeted therapies. So the big area of predictive pathology is finding out what are the genetic lesions in a tumor and how will the tumor respond under therapy and change to resistance is becoming more and more important in larger and more patients. Now coming back to diagnostics, the problem we have in anatomical pathology is that we frequently deal with very small biopsies and very little tissue. And we need, of course, need a correct histology. We need to know that this is a carcinoma of the lung, not a metastasis from another carcinoma, not a lymphoma or some other disease. So we need to have histology, immunohistochemistry, and then determine all those predictive biomarkers to guide therapy. So and these biomarkers are growing more and more and more every month. So our idea was to step back from testing single biomarkers and single genes and rather go for testing all genes at the same time and finding out even the small subgroups of patients that qualify for specific therapy. And the idea that has now become very practicable in many labs throughout the world and is also commercially available is to select an informative gene set which you need, for example, to treat lung cancer patients, or let's say a melanoma set for melanoma patients, that includes all biomarkers, all genes that you would like to sequence and analyze for therapy. And then from these little slides with very few cells, extract DNA from formalin-fixed paraffin-embedded slides, quantify your DNA, and generate a library with all these genes for PCR, and actually can multiplex this for many different patients by medical identifier. So you know this ACT, exon 20, for example, PCR is generated from Mrs. Miller, and that ACT, other ACT is generated from another patient. And then sequence them with next generation sequencing technology at the same machine, at the same run, multiple patients, multiple genes, and very high density to go in a very deep. We sequence routinely 5,000-fold coverage. So every PCR product is sequenced 5,000-fold to detect with high sensitivity even very few cancer alleles in the background of normal cells. And then this is analyzed through a bioinformatic pipeline, and then it is transported into our routine diagnostic storage, where all the histopathological data information is stored, and then it's annotated what genes have been sequenced, what is the modality of the gene? Is it activating the kinase? Is it inactivating? And what's the allele frequency? And is there a study available that would be a perfect study for this specific genotype and disease? So the bioinformatic pipeline is actually very important to handle this massive number of sequences from large number of patients. And as you can see, I've brought here a beautiful example of a patient, actually our first patient who we identified as being lung cancer adenocarcinoma with a ROS receptor gene fusion that's kinase activating. And she initiated at very late stage 4 therapy with crizotinib 2.5 years ago and still in complete remission. So some of these genetic subtypes of lung cancers are very, very sensitive and vulnerable to specific therapies. And since lung cancer is the cancer type worldwide with the highest burden of mortality, even small subgroups like ROS actually is 1.8% of all lung cancer patients. Adenocarcinomas in our center, these are not rare patients. These are big numbers of patients worldwide. And even rarer mutations can be detected by these genomic approaches, like we've recently described a novel fusion between neuregulin, which is the ligand for HER1, HER3, HER2, and the pan-HER activator. And also this fusion is vulnerable to inhibitors of pan-HER signaling. And the HER signaling pathway is activated, and you can show this in tumors by hyperphosphorylation, and the cell lines, appropriate cell lines, are sensitive to inhibitors. That is not only true for lung cancer, because more and more diseases, more and more neoplastic diseases become a subject of intensive genomic analysis. I'll just briefly show you CLL, chronic lymphocytic leukemia, which is the most frequent leukemia in the Western world. And it's characterized by a very, very diverse biological and clinical course. There are patients that are stable for a long, long time, for many years, and in the first years practically need no treatment. And there are subgroups of patients that have a very aggressive form of CLL, which need early treatment, and it is important to classify the patients according to their risk score. And so we started out and analyzed all genes that were reported from the big international genome consortia for CLL, which is genes in the B-cell receptor signaling, in the Notch receptor signaling, Toll-like receptor signaling, also genes that signal DNA damage like P53 and ATM. And we immediately realized these genes tend to be very, very large, like ATM has 117 exons. And some exons are so large that you need multiple PCRs from FFPE material. So there was no chance to do this by multiplex PCRs. And so in this case, we went and applied an enrichment technology where we enrich all informative genomic areas and then sequence it. And thereby we generate a massive number of sequences. We found in 136 patients more than 20,000 variants, which were filtered by an informative frequencies. And then they were filtered to remove all synonymous non-protein changing variants. And then we filtered for the typical formalin artifacts and filtered out SNPs, known SNPs. And then we remained with only 173 variants in these patients. And by this massive reduction of variants, you can see that it is very important to have a backbone of a very, very rigorous bioinformatic pipeline, because you are not able in a diagnostic setting to analyze 20,000 variants by hand and eye. What we found is that by our genomic mutational type, we could easily classify those patients with a high-risk CLL that needed early treatment. For example, the known parameter P53 mutation was highly enriched in these patients. And on the other hand, the majority of patients that had a more benign clinical course and needed no early treatment. Molecular staging. It's a big question if you have patients with multiple tumors, what is the metastasis and what is the second tumor, because that heavily influences clinical decisions. And I show you just briefly 2 cases I teach for the International Academy of Pathology. Here we had 2 tumors of an invasive pulmonary adenocarcinomas in segment 2 and 9. And the question was, is this a metastasis, a primary and a metastasis? In this case, it was initially staged as clinical stage T4, not undergoing resection, and also with a lymph node metastasis. And so we were asked only the question, is there an EGFR mutation? So what we did, we analyzed our gene panel and found that every tumor in segment 2 and segment 9, and also a small adenocarcinoma in situ, which was not diagnosed previously, had its individual molecular signature. So we had actually a patient with 3 different very small tumors, and all were surgically removed, because this patient is now not a T4 stage patient, but a T1A with 2 tumors, multiple tumors. So this really influences the standard diagnostics and the clinical decisions of what patient to surgically resect and what patient to render to chemotherapy. Another patient was a patient with 2 neuroendocrine tumors in the lung. Then he was staged, and they found a second tumor. This is the lung tumor. Looks like a very aggressive neuroendocrine carcinoma. And the gastric tumors at first glance look a little bit similar. And both tumors actually express neuroendocrine markers. So we went to genomic sequences and found that indeed the patient had 2 different tumors with 2 different private P53 mutations. Only one tumor had a P10 truncation mutation and the other had a MET mutation. So molecular profiling really can tell you the molecular evolution of the disease, also according to standard anatomical classification from T1 to T4, and to discriminate metastasis from multiple primary tumors. And this is a list of genes that have a very different mutational spectrum and are useful to classify private mutation in individual tumor lesions over time. And surveillance, this has also been a question more recently. Can we monitor these mutations also in the blood of patients? And indeed, this is a very beautiful example, a typical exon 19 deletion mutation in exon 19 of EGFR. Patient received Tarceva, and in the blood we found this T790M mutation. If you remember my first patient, so we don't have to rebiopsy these patients, but we can detect the mutations in the blood. However, this is a very sensitive technology. If you analyze these patients too late and keep the blood for several hours on room temperature, then as you can see here, there is no mutation anymore in the blood, but it's still present in the tumor. So it's a sensitive technology which we need to learn and to practice in clinical applications. Now, there is now very good reference databases, actually by the Ingenuity pipeline, to analyze large collections of tumor lesions. For example, by the National Comprehensive Cancer Network, by the TCGA consortium, by the Cosmic Library. You can filter out for every tumor you would like to analyze, like ovarian, lung, liver, BRCA genes, and so forth. What are the genes that are interesting for my clinical questions? And then you can either design your own PCR pipeline, or you can use a number of already commercially available mutations, like for example this Actionable mutation panel from QIAGEN, and analyze these genes in your tumors. And this, I've shown you the work pipeline from FFPE slides, extracting DNA, generating small libraries, adding medical identifiers and sequencing primers, and then go on a next generation sequencing platform and sequences with high coverage and high sensitivity. And such panels are now available for many tumor applications, so you don't have to sort of dig in these public libraries and search for informative genes. There are commercially available gene sets adapted to breast cancer, to colorectal cancer, to lung cancer, and so forth. And we have tested many of those, and indeed they have very high coverage with very high specificity, so you get really the sequence information on the exons and the genes that you have selected in your panel. So to summarize this, I think pathology is currently really changing by integrating genomic information into histopathological tumor diagnostics. We need to classify better tumors by adding genomic information. In sarcomas, in many instances, the specific genomic lesions are defining the entity, also in lung cancer, as I showed you. In breast cancer, it's now applied to subclassify the types. We need these sequencing informative gene panels to predict responses to therapy, to come away from empirical therapies, to find out and predict who will be a good patient for the correct drug. We need these panels to come to a better prognosis of the clinical course, to decide in many instances, for example, CLL, that no therapy at the moment is the best therapy. We also need it for better tumor diagnostics, to monitor the to survey the evolvement of resistance and relapse, but also to decide with very high accuracy what is the metastasis and what is the primary tumor and what is a secondary tumor during the course of a long-term survivor of cancer. And with this, I would like to thank you for your attendance. I'm here to answer all your questions. And I have to thank many people in the network of genomic medicine. And if you Google an NGM, lung cancer, Cologne, you will find all these people and all their data. And I'm thankful to work in such a fantastic pathology group. Thank you very much.
Thank you, Reinhard. As a reminder to attendees, you can type a question into the Q&A box at any time. We will now turn the webinar over to Vikram Devgan of QIAGEN. Please allow us a moment as we hand the webinar over to Vikram. Vikram, please begin whenever you're ready.
Thanks, everyone. First of all, I would like to thank Professor Büttner for providing such a wonderful presentation. You have already heard all the real-life example of giving what are the applications coming out of QIAGEN GeneRead panels, and not only the panels but also the applications of QIAGEN sample to insight next-gen sequencing workflow. So what I'm going to do today is I'm going to talk more, basically follow up what Professor Büttner has said. And I'm going to talk more about targeted DNA sequencing. What are the challenges associated with the implementation of targeted DNA sequencing into the clinical settings? And also basically reinstate all the QIAGEN solutions which Professor Büttner has already highlighted in his presentation. How we are using those QIAGEN solutions to address these challenges in the clinical research settings. So, okay, first of all, I would like you to note that all the products which I'm going to mention in this presentation all are meant for molecular biology application. They are not intended for any diagnosis, prevention, or treatment of disease. So having said that, let's get into the presentation now. As you all know that the DNA is the basic unit of genetic information, and any change in DNA sequencing can lead to the physiological or phenotypic change, and these changes can vary from single nucleotide mutations to insertion, deletion, to copy number change, to fusion genes. And it's very important to detect these mutations. Not only they play as diagnostic and prognostic markers, but also help us in selecting proper treatment for our patient. So let's take an example of BRAF V600E. It's present in almost 60% of melanoma patients, and if you look at into the drug, the PLX4032, it will be beneficial in melanoma patients only if they have the BRAF V600 mutation. So that basically shows the importance of our need to detect these genetic variations. Now, if we look into the technologies for detecting these mutations, that has been evolved over the time. So you must remember about the ARMS PCR method, which can detect the few mutation at a time. Then came liquid bead arrays, followed by sequenom mass arrays, and then came the next-gen sequencing, which basically has revolutionized the way we are extracting genetic information from the clinical samples. It basically provides us the unique ability to detect multiple mutations in a multiple genes with a multiple samples with a single next-gen sequencing run. So that's really remarkable ability to do that, especially for clinical samples. However, if we look into the DNA sequencing or DNA next-gen sequencing, there are 3 different flavors. So one can go for whole genome sequencing, whole exome sequencing, or can be the targeted DNA sequencing. Now, if we look into which of these are best for clinical utility, obviously the answer depends on what you really look at. But if I have to give the analogy of these sequencing, I'll say that the whole genome sequencing or whole exome sequencing is more of the high-level view, whereas targeted DNA sequencing is looking into the more detailed or more granular view of anything you are looking at. By doing a target DNA sequencing, you can really focus on the region or a genomic region which is clinically relevant. And doing that, you are going to generate the data, data output is going to be more relevant for your clinical use, as well as the cost of running the sample or analyzing per sample is going to be low. And importantly, if you are dealing with tumor samples, we know that there are heterogeneity issues associated with tumor samples. In that case, driver mutations are generally present in very low mutation frequency. And in that case, to detect those low mutation frequency, you need to do more in-depth sequencing, or that can be enabled only by targeted DNA sequencing. So that's another big advantage of doing a targeted DNAseq. And obviously, if we are dealing with the clinical setting, the samples are going to be very precious, and the target DNA sequencing needs only a very low DNA input, ranging from 100 to 10 to 250 nanogram. So taken together, we can say that targeted DNA sequencing is so-called the best choice for clinical utility. So, and target DNA sequencing is available in the market from quite some time. It's not new at all. I mean, you already seen a lot of application in Dr. Buttner's presentation. But if you really look into that, there are still a lot of challenges associated with it when we are trying to implement in a clinical research setting, starting from DNA quality and quantity. Most of the FFP samples are circulating free DNA or fragmented DNA. And it becomes very important to, you should do accurate quantification of your DNA, which is so-called accessible to target enrichment. Then for the clinical settings, we need a quick turnaround time. The 4-day from sample to result is so-called more acceptable nowadays. And then let's not forget about data analysis and biological interpretation. This is still a bottleneck for next-gen sequencing. And to overcome these challenges, what we require is, first of all, we require the ability to quantify functional DNA. And when I say functional DNA, what I meant by that is the DNA which is accessible for target enrichment. And then we need a technology for DNA sequencing which is rapid and can be compatible with the degraded samples. Let's say for the FFP and circulating free DNA samples, which have on average the DNA size of 150 base pair or so. And above all, we need to have easy to use data analysis tools. So these are the requirements to overcome these the challenges which are listed above. And keeping these requirements in our mind, the QIAGEN has developed the targeted DNA sequencing solution. We call it GeneRead DNAseq targeted panels. This panel uses basically a multiplex PCR-based technology. And what we have done here, we have designed primers all over the genome. So any gene, any exon, or any hotspot, or any region in a genome which you are interested, we have primer designs for that. And then you can pick and build any your panel of which is really unique for your needs. And in all these amplicons, the average size is around 150 base pair, which make them very compatible for all the samples, whether we are talking here for FFP sample, liquid biopsy samples, or single cells. So this target enrichment panel is compatible for all of them because of its unique small amplicon size. And then the input DNA requirement is only 10 nanogram per DNA pool so that you can save for your precious sample. It takes only 3 hours to do a target enrichment and thereby enabling to go from sample to insight within 4 days. And to give you the best specification, what we have done, the genes, the primers which we have designed for each gene, we have segregated them in 4 different tubes. So although you are going to start with 4 different tubes, but you need only 1 library preparation per sample, so that will simplify your process or your workflow. But most importantly, these gene panels come as an integrated part of the whole standardized workflow from sample to insight from QIAGEN, which has integrated data analysis part. We have CLC Cancer Workbench, as well as the data interpretation part from Ingenuity Variant Analysis, HGMD, and Ingenuity Clinicals. And this whole workflow is completely standardized so that you can spend time in linking your biological interpretation or clinical interpretation to the clinical significance. And QIAGEN has taken care to do all this standardization so that you don't have to spend time in standardizing your target DNA sequencing workflow. It has already inbuilt QC steps to ensure that you are going to get a good result with each and every run. And right now, what we have the largest collection of wet bench validated gene panels in the market. So we have 14 different panels, and all ranging from different needs, all the way from solid tumor, very focused panels, to hematologic malignancies, to tissue-specific panels. You name the cancer, and we have the panel for it, as well as comprehensive cancer panel for carrier testing and cancer predisposition panels, so on and so forth. And a lot of work has been done to ensure that the genes which we are selecting in these panels are clinically relevant. So we have spent a lot of time going through all different databases like TCGA, NCCN, COSMIC, CleanVar, as well as our proprietary Ingenuity knowledge base to ensure that we are picking the genes which are highly or the most relevant for your specific cancer type. But we also understand that the one size doesn't fit all. And we have, in that case, we have an ability to provide custom panel builder. So we have an online custom panel builder, which you can go online on our QIAGEN.com and put any genomic region or any gene, and you can design the panel specific for your need. So that was about the panels, but to have the really good target enrichment using those gene panels, it's very important to have proper or accurate quantification of input DNA. Otherwise, it's like garbage in, garbage out. And when you're dealing with the FFP samples or circulating free DNA, due to the degradation, it's very important to ensure that we are doing an accurate quantification of the DNA which is accessible for target enrichment, as inaccurate quantification of input DNA can lead to the poor target enrichment, poor library yield, and eventually you'll waste your whole sample as well as time and money because you're not going to get any reproducible result out of that next-gen sequencing. So to overcome that challenge, what QIAGEN has done, we have developed a GeneRead quantumized system. And what it does three things with a single qPCR reaction. First of all, it do the quantification of PCR amplifiable genomic DNA. Second, it provides a quality control score for all the DNA based upon degradation status and the contamination. And the most important part is it provides you the guidelines to rescue your low quality DNA. Because if you are dealing with the clinical samples, the sample is precious. Although it's a low quality, you would like to make use of it and you want to see if we can get some reliable result out of it. And that's what this kit does. It will provide you the guidelines by optimizing number of PCR cycles for target enrichment so that you will get the good result with each and every next-gen sequencing run. And how does this kit work? So it basically uses a simple qPCR reaction. What you'll get is you'll get two qPCR assays. With one is for 100 base pair amplicon, another is for 200 base pair amplicon, along with the control sample. And what you have to do is just run qPCR using these primers along with the control sample and your case sample, and then take the CT value and upload on our online data analysis tool. It will do all the calculation on its own. It will look into the delta CT value ratios, and at the end provide you quantity and quality of the amplifiable DNA, which will give you the assurance that each and every sample moving forward is going to give you the reliable results. Now, to make sure that these gene panels are applicable in a clinical setting, it's very important to do some, the initial verification. Like we have to look into the accuracy, sensitivity and specificity, precision, and reproducibility. And we have done these for all the 14 panels we have. So ideally, you should do all these three things with each and every panel. But since we were dealing with 14 different panels, what I'm going to do is I'm going to show you the few representative examples of different panels. And for each of these experiments, we have used our universal workflow from sample to insight, which is shown here. So we have we have used all our QIAGEN products ranging from sample isolation to target enrichment and library preparation. And these all experiments has been run on MySeq platform. As I said, that our sample to insight workflow is compatible with any next-gen sequencing sequencer. So let's look into all these tests one by one, starting with accuracy. So to test the accuracy of our panels, what we have done, we have picked BRAF V600E mutation and selected 10 different clinical specimens from different cancer types, and they are known to have this mutation as different frequency. And then we tested that using our targeted DNAseq gene panels, and here are the results. As you can see that all the mutations has been accurately detected by our tumor actionable mutation panel. So it shows 100% accuracy in calling BRAF V600E mutation. And ideally, you should do this for each and every mutation which you are going to report at the end of your experiment. This is just a representative example for BRAF V600E, but you are supposed to do for each and every mutation which you want to report at the end. The next, for sensitivity and specificity example, for this testing we used NIST genome in a bottle genomic DNA. So for that we used different dilutions of NA12878 genomic DNA in the background of NA19240 sample. So we used different dilutions ranging from 50% to all the way to 10%, going to detect the 5% allelic frequency. But for the sake of simplicity, I'm going to show here only the data from 10% dilution. And in this case, we have used carrier testing panel, which covers around 663 kilobase genomic DNA. This targeted region had 240 SNPs and 14 indels. With the 75% of these are heterozygous in the nature, so we are looking at 5% allelic frequency. Now, if you look into the results with the whole universal workflow, all the way from sample to result using QIAGEN workflow, we get the sensitivity of 87% with 5% mutation detection. And the false positive rate is around 42 base pairs per megabase. And in case of indel, the sensitivity is 82%, with a false positive rate is 92 per megabase. And you have to remember that here we are talking about the ability to detect 5% mutation frequency. As you go toward the higher mutation frequency, the sensitivity will improve as well as the false positive rate will decrease further. And with the 5% mutation frequency, these sensitivity and false positive rates are pretty good with any kind of comparison. And for next to look into the precision and reproducibility, what we have done, we have took the control sample, which contain a wide range of endogenous genes, ranging from EGFR to NRAS, BRAF, KRAS, and all of them have mutation at different allelic frequency, ranging from 24% to 1%. And we tested these control sample in 3 different labs using in triplicates using 3 different lots of our clinically relevant tumor panels. And the data is shown here. As you can see that in a triplicate and with the 3 different labs, and also using even with the different lots, we are getting very reproducible results over here with very high precision, and which, as shown over here, till the limit of detection is more than 10%. However, if you look into the mutation which are less than 5%, we are not able to call them using our online data analysis tool. But this is the reason we are behind not able to call this is because the mutation is less than 5%. In the another sample, the similar mutation, if they are present more than 5%, we were able to call them using our sample to insight solution. So basically, after this test, we have ensured that we have a high accuracy, sensitivity, and specificity, as well as precision and reproducibility. After confirming this, we went ahead and did a short case study in which we have used our colorectal cancer panel to do the screening in colorectal neoplasma FFP samples. Just to give you a small little background over here, as most of you must know that the colon cancer is the 3rd most common cancer in U.S. And 40% of the patients has the KRAS mutation associated with this tumor. And if the patient has the KRAS mutation, it's unlikely to get benefited by EGFR inhibitors. So what we have done here in this case study, we have used our GeneRead colorectal cancer panel to screen 21 randomly selected colorectal FFP samples, which are previously tested through sequenom mass array. And here are the results. So I would like to show you the results starting with the GeneRead Quantimise kit. This is the kit, as I mentioned before, you can use to do the quantification, checking into the quality, as well as optimizing the PCR cycles. So you can see very high quality of all the samples, and it shows the input quantity does vary from sample to sample, which is quite expected because we are dealing with FFP samples. But not only it can do the quantification and provide a quality score, it also can tell you how many number of additional cycles you need to ensure the reproducible results at the end. So as you can see there that it's basically splitting the samples into 2 batches, one with the 7 additional samples and another with the 9 additional PCR cycles. So that's exactly what we have done. And here is the final results. As you can see here, the 12 out of 21 samples has shown the mutation in KRAS at codon 12 and 13 with the different mutation frequencies. And whereas the 9 of them seems to be wild type. So just to basically quickly summarize this whole thing, we shows that this case study shows the 55% of colorectal neoplasma FFP sample has mutation at codon 12 and 13, and the result from GeneRead colorectal cancer panel is they were in 100% concordance with the sequential mass array results. However, importantly, although the results are in a completely 100% concordance, the important part over here is by implementing or by using this targeted DNAseq gene panels, the customer has saved their precious sample as well as time and money as compared to the sequential mass array. And then finally, the quantumized kit has ensured the successful NGS result with all the FFP samples. With that, I would like to conclude this seminar, and would like to take any questions you have.
Thank you, Vikram. We will now enter the Q&A portion of the webinar. Our first question for Reinhard Buettner. Can you comment on resistance in the clinic and how NGS applies to that? It seems that mono or sequential therapies are still going to ultimately result in recurrence of resistance. How does NGS apply?
Yes, and in most of the cancer types, especially in solid cancers like lung cancer and sarcomas, we are talking about treatment of a systemic disease that will prolong the life of the patients, but it will not mean eventual cure. So you do have to monitor the emergence of resistance, especially since we now have several tyrosine kinase inhibitors, several modalities of treatment, combinations with chemotherapy, switch to an immune checkpoint therapy at hand. So we have many, many therapeutic options now. And at every stage, when we make a new therapeutic options, we have to recheck the mutations and make the decisions what additional genetic mutations require what therapeutic steps. So we are not talking about curing cancer, we are talking about bringing cancer into a chronic disease stage and applying sequential, targeted, and for this patient, selective therapies. And therefore you need always to analyze informative gene sets.
Thank you. Somewhat related to that question, do you know what impact the exon 20 inserts, around 5 to 10% in the EGFR gene, have on TKI treatment response? Are they predictors for resistance overall?
Well, it's a difficult question, and it shows that even such a good old gene like EGFR, which has been probably sequenced for 100,000 times all over the world, poses new questions in daily histological questions. I think there are different types of insertions, and a good data library to go to is the COSMIC library and also the CBio portal from Memorial Sloan Kettering, the Chris Sanders library, and check, there's information on the modality of the informations. Are they sensitive to TKIs? And if I have a diagnose a complex exon 20 insertion, I tend to go to recommend treatment with the new third generation TKIs because they tend to be not very sensitive to the first generation like Erlotinib or Gefitinib.
Thank you. This question is also for Reinhard Buettner. Based on what level of evidence do you make a decision to use a gene in the diagnostic panel?
That is also a tough question, and life is more than black and white, and there are intermediate gray shades. So we tend to classify our pathological report into 3 categories. There are clearly mutations where there's a lot of clinical information that they sensitize a tumor to a specific treatment, or they confer a primary resistance. Good example would be an activating KRAS mutation in the classical hotspots areas like in exon 2 or exon 3. And then these tumors do not benefit from EGFR treatments, both in lung or in colon. And then there are mutations where we have amber clinical data that they sensitize tumors, like a BRAF mutation in melanoma or a typical exon 19 deletion in EGFR. So these are the bona fide where we report these are activating or they're inactivating, they confer sensitivity or resistance. Then we have clear SNPs where there's good data that these occur in the germline, and actually in many instances we tend to check also some non-tumor DNA tissue, DNA extracted from non-tumor tissue. And if it's there also in the germline, let's say a missense amino acid changing, then we report it's probably a SNP. And there are good libraries now where SNPs are recorded and you can check for them. And then there's this intermediate category that also is known in human genetics, unknown variants. We have a lesion, but we do not know what's the meaning. And as pathologists, we are used to make clear predictive diagnostics with high level of security. We would recommend treatment on the secure, safe mutations with known clinical experience. And the unknown variants and the SNPs are not useful to make a clinical decision for a specific cancer therapy. So there are these 3 categories.
Thank you. Our next question is for Vikram Devgan. Vikram, even after enrichment, do you see degradation like Dr. Buettner showed in primary samples?
So when we are talking about degradation after enrichment, there is a cleanup step, and which will get rid of all those, the so-called degraded samples which are getting carried over after the target enrichment. So we do have a purification step after the target enrichment, which can get rid of all these degraded samples.
Can I add one sentence to this?
Please go ahead.
If you see that you lose, for example, through the purification step, a lot of DNA, probably the most likely reason is there's a lot of degradation. The typical reason is overfixation or heavy underfixation, and the tumor is not fixed well and it degrades because all the enzymes still work. And then you, as a pathologist, you should talk to the primary people that handle the tissue. I think you need to improve the tissue fixation. That's a very important step.
Sure.
Thank you. Our next question is for both panelists. It's a 2-part question. Could you also comment on how your patient work with NGS is reimbursed or financed? And related to that, what's the average cost of the panel without the interpretation?
Is that a question for me, Ben?
I believe that the first question is targeted more toward you, Reinhard, commenting on how the work with NGS is reimbursed or financed.
Well, there cannot be a single answer to this because in different countries there are still totally different reimbursement systems that range from no reimbursement at all to now ways that will eventually lead to a good reimbursement system. All I can say, what we have done in Germany now is that we talked to the health insurance companies, and they made for certain comprehensive cancer centers, they make individual contracts that will reimburse analyzing panels of genes. So this is not being charged from the clinicians, for example. I think it's a good model to separate the reimbursement of testing from the reimbursement of treating the patients. I know in many countries there's no reimbursement at all, and then usually pharmaceutical companies pay for it that want to enhance testing, to enhance usage of their drugs. And then there are some individual health insurance companies. I think until last year, Germany prohibited next-generation sequencing from routine diagnostics, and it has changed now with the reimbursement of packages and panels.
Thank you, Reinhard. We must conclude the webinar at this point in time.
Ben, just to answer that last part, the cost of the workflow for if we not include the data analysis part, depending upon which panel you're using, the costs range from $100 per sample to $125 per sample.
Thank you. We'd like to thank our panelists, Reinhard Büttner of University Hospital Cologne and Vikram Devgan, head of the Biological Research Content Business at QIAGEN. If we didn't have time to get to your questions, we will try to have the panelists answer them directly afterward. In addition, please take a moment to complete the brief survey that should pop up on your screen before logging off from the webinar. Your feedback is extremely valuable. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to attendees. Thank you for joining us for this genome webinar.